we call a protein. Okay? And now I think we're cooking with gas. It's looking
the
good.
same slide? Here, hold on.
Yeah, this is cool.
Okay.
Great. All right, so thanks, guys. So we link together these amino acids to make proteins. This is the structure of an amino acid. Okay? It's the basic monomer, the fundamental unit associated with proteins. And all amino acids are going to adhere to this basic general structure. There's 4 or 5 parts. Let's talk a little bit about each of them. You guys recognize this one over on the left right out of the gate, right? Because this is one of the 6 or 7 functional groups of carbon that we've talked about. That's an amino group. It's shown here sort of in its neutral state, but at pH 7, like what you would find inside of a typical cell, it's going to have a positive charge associated with it. Amino groups typically have a positive charge associated with them. How about that? All right, so we're going to have something positively charged over here. On the other end of the amino acid, you recognize this, right? The screen's shouting it out at you, but I think just from the structure alone, you'll probably know what's going on. This is another one of those 6 or 7 functional groups of carbon. That's a carboxyl group. At neutral pH, carboxyl groups typically have a negative charge associated with them. So check it out. Amino acids have sort of like this schizophrenic personality, right? On one end, they're positive, they're upbeat, right? On the other end, they've got this negative charge from the carboxylic acid. They're kind of a downer, right? One half of the molecule is acting, it's behaving, it's reacting exactly the opposite as the other half of the molecule. And I think you can see right out of the gate how it is that one amino acid might be particularly well suited to satisfy the needs of another amino acid. All you need to do is rub the carboxyl group of one amino acid up against the amino group of the other amino acid, and you're on your way to establishing a bond that links together the 2 amino acids, right? They're going to have a natural affinity for each other because of those opposite charges attracting. Straightforward enough, right? So when you look at the structure of amino acid, all of them have this amino group associated with them. All of them have this carboxyl group associated with them. And to sort of keep peace, I suppose, but also to provide something to dangle other things off of, all amino acids will also have this central carbon. If you want to be really fancy, you want to impress people walking down the halls in the biology department building or the biology building, go up to somebody and say, oh, yeah, I know all about alpha carbons, right? Alpha carbon is the carbon in the center of these amino acid structures, right? Frankly, I don't try to impress people by talking about alpha carbons. I'll just say it's that center carbon, right? It's all the same thing. People will know what you're talking about. But there's a carbon in the center from which these other things get attached. One of the things that gets attached, the amino group. Another thing that gets attached, the carboxyl group. Another thing that gets attached is, dealing with all the boring parts first, the hydrogen. All right? Every amino acid off of that central alpha carbon has a hydrogen dangling off of it. Right? What's up with that? Does that say something to you? I don't think it should say too much. If any, I think what it should say is that's just sort of like a space holder, right? It's filling up the gap. It's helping that carbon be happy. Its valence is full, right? 4 covalent bonds off the carbon. That hydrogen's not actually really doing anything. I think it's unrealistic to be thinking about it in terms of storing energy. I think it's unrealistic to think about it in terms of being hydrophobic. It's just too small, right? It's all of those things, but just not in any big way. Bottom line, you got a central carbon. There's something important happening here that's going to help you make a bond to another amino acid. There's something important happening here that's going to help you make a bond to another amino acid. There's something happening here that rounds out the valence of the carbon, and the action is over here. It's the thing that dangles off of that other bond that the carbon in the center needs to have filled. And that thing is called an R group, right? R is short for reactive or reaction group, right? That's ultimately where the real action is for the amino acids. And you're wondering, I suppose, some of you at least, R, is that the name of, is that a one-letter abbreviation for an element, right? H is hydrogen, C is carbon. What's R? Is that argon? It's not. Argon is AR. What's up with R? R is shorthand for reactive group, and life on this planet uses one of 20 different structures for that R group, right? So, before you guys really started to take this class, I asked you a question. I asked you a bunch of questions. One of them is this fourth question, where I said, how many different amino acids do we use? I think I said, made it very personal. How many do humans use, right, to make our proteins? And I'll tell you, right now, I hope most of you are gravitating toward B as the right answer. Because I just said there are 20 different R groups. That means you can make 20 different amino acids. And happily, even coming into this class, before I said a word to you, effectively half of you knew that, right? Some of you bit on 4, right? And I'll tell you why I chose these particular numbers. These are not random numbers that are in there. I was hoping I could get some people to bite on 4. Maybe not hoping, but I was thinking this might be a plausible answer, because there are 4 different nitrogenous bases. There are 4 different kinds of nucleotides. I'll talk with you about that at the second half of today's lecture. That's about DNA, all right? So there are 4 of them. There are 20 different R groups, which give us 20 different amino acids. There are 64 different triplet codes. That's where that number came from. We'll talk more about that later in the course. And there are 3.2 billion nucleotides that spell out the instructions of the human genome. And again, I'll talk with you more about that in the second half of the class today. But the answer to the question was, there are 20 different amino acids that we use to build all of our different proteins. And what makes one amino acid different from another amino acid is very simply what's going on over here, all right? So that R is shorthand for 20 different structures, 20 different chemical structures that you can dress these core parts of the amino acids up with. Each of the 20 different amino acids has a different R group. Okay? Get ready. Maybe you get a little adrenaline going here, because in the next slide, I'm going to show you at one time. I don't know, I was tempted to ease you into it, going 1 or 2 amino acids at a time, but I'm going to just throw it all right at you. The next slide shows you all 20 amino acids in all their glory. You ready? All right, brace yourself. Nobody's looking as excited. There are a couple people who look excited. All right, here you go. I should have the 2001 Space Oddity music playing right now, because there they are. All right, should I just pause, let you guys catch your breath a little bit here? I know it's a lot to soak in. All right, those are the 20 different amino acids. All of them, all in all their glory. There's parts of these amino acids that are the same, right? All 20 of them have this same purplish thing associated with them. You know what that purplish thing is? It's that amino group. It's that central carbon with its hydrogen dangling off of it, and it's a carboxyl group. Boring, right? Compared to what's going on with the R groups. That purplish thing is the same for all 20 amino acids. Each of the 20 different amino acids has a different R group. The yellow, green, pink, or blue thing that is on the rest of these structures. Okay? Each of those structures is different. So, I know we got a weekend coming up, and you're probably looking for something really exciting and interesting to do. I'll bet a lot of you are starting to pencil into your calendar the idea that I got to set aside some time to memorize the structure of all 20 of these amino acids, right? Chomping at the bit, right? Well, you know, I'm toying with the idea. Yeah, go ahead, do it. That'd be great. Good for you. Have fun with that. But I don't think it's necessary for this class for you to actually memorize the names and structures of all 20 of the amino acids. Okay? So I'm not going to ask you to do that. I'll tell you this. Over the course of my professional career, I have probably had to commit to memory those names and structures maybe 20 times, right? The first time I had to memorize them, I remember it was in a freshman biology class, and it took me a good 5 hours. Maybe I'm a little slow, right? But I had to stare at these things for like 5 hours and quiz myself on them. It wasn't a particularly pleasant experience, as I recall, but nonetheless, I did what had to be done. I memorized them, and I moved on. And I promptly forgot those names and structures within a week or so of needing to know them for a test. That's just the way I am. I suspect I'm not that alone in this room in saying that you'll forget them pretty quickly. But there's good news, because the second time I had to memorize the names and structures for another class of these amino acids, the second time, it only took me about 2.5 hours, about half as long as the first time. Something apparently had stuck. And the third time, it only took me about an hour. And the fourth time, it only took me about half an hour. And the 20th time, sometime last week, took me about 2 minutes. Right? So the more times you have to memorize these names and structures, the easier it becomes. And I can guarantee you this, if you take the second course in this series, starting in the spring semester, mean old Dr. Goldstein and mean old Dr. Cipollini are going to insist, they'll be appalled that I didn't make you memorize those names and structures. So if you got some time this weekend, put in a down payment. It'll make your life easier next semester, and the semester after that, and the semester after that. Right? Because at some point, many people are going to insist that you memorize the names and structures of these amino acids. But I'm not going to be the guy who does that. Please remember that at the end of the semester during the student evaluations of instruction. Right? I'm doing you a solid. Right? You know, let's just reciprocate here a little bit. I'm not going to make you memorize them. But here's the thing. I am going to be as insistent as I can that you know what's up with each of these 20 amino acids. I want you to know that basic structure, the amino group, the carboxyl group, the alpha carbon, and the hydrogen off of it. And I want you to appreciate that the amino acids can be parceled out into 4 different categories, different flavors, different things that are on the palette, again, that you can paint with when you're designing, when you're building a molecule that can get something done, a protein, right? I want you to know there are 4 basic different kinds of amino acids. I want you to also be able to say, gee, that's a plausible structure for an R group, and that structure would fit into this category as opposed to that category. And beyond that, I want you to also have this appreciation that within each of the categories, there are some differences between the R groups, right? And those differences almost always come down simply to, ooh, that's a big one, or, oh, that's not so big, or, oh, it's kind of in between, right? It's size related, right? So let me say it to you plainly. You don't have to memorize these structures and the names of the different amino acids. I do want you to realize there are 4 different categories of amino acids, and be able to put a structure that I give you into one of those 4 categories, because that's something that I hope you'll remember for the rest of the time you think about proteins, however long that might be. And just beyond that, appreciate, oh, some of these are relatively small structures, some of them are bigger and bulkier. That's a minor point, but it's a useful one to have in the back of your head. So there are 4 different kinds of amino acids. There are the ones where the side group, the R group, is hydrophobic. There's the ones where the R group, the side group, is hydrophilic. You guys know what that means. Wants to stay away from water, wants to hang out with water. And then there are those where the side group, the R group, is not just hydrophilic, it's flamingly hydrophilic because it is negatively charged. And those where the side group is not just hydrophilic, it's flamingly hydrophilic because the side group is positively charged. 4 different kinds of amino acids. We'll talk a little bit about each of them.
Yeah, I was just curious, if, like looking at the last slide, it has 2 hydrogen attached to the nitrogen, and I was just wondering, like, it says H3N+. Is there a reason for that?
Well, what I was saying when this was up here a little while ago is that at neutral pH, you would expect this to have a positive charge and this to have a negative charge. Now, there's been a sleight of hand. Nothing else has changed. It's just now we're talking about neutral pH. And so on this diagram, you're seeing that +1 on the amino group and the -1 on the carboxyl group. So here are the 20 amino acids. I'm going to talk with you about each of the R groups a little bit and tell you why it is that that makes it fit into one category or the other. And then we'll talk about how you string these guys together and do something with them. So let's get the hydrophobic guys out of the way first. They were at the top of that last slide. Let's zoom in on them and see what we see when we're talking about the hydrophobic amino acids. Just keep this all in perspective. There are 20 different amino acids. Some of them have an R group that is hydrophobic. Do you need me to say anything more? I'm afraid I probably will, but I'm inclined to just walk away and say, you don't need me anymore. You can figure this out on your own. Look at what's shown in the yellow boxes here. That's the R group. What do you see when you look at the things in these yellow boxes? I hope that what you see is nothing, and I mean nothing, but nonpolar covalent bonds. Stuff that water's not going to want to have anything to do with. There's nothing that water wants to rub its slightly negatively charged oxygen up against. There's nothing that water wants to rub its slightly positively charged hydrogens up against. Water is completely disinterested in the stuff that's going on in the yellow boxes on this slide. That's why these R groups are hydrophobic. They're afraid of water. They run away from water. They hide from water. Water doesn't seek them out. Water could care less about them. They are dead to water. All right? Water has no interest in these things. Every one of these bonds, every one, nonpolar covalent bond. Nothing but carbon to carbon and carbon to hydrogen bonds there. Do I need to say more? Unfortunately, I think I will, but I think I could probably stop. The only other thing to look at here is the fact that, oh, look at that, glycine. That's the name of one of these 20 amino acids. All of the amino acids end in "ene," by the way, all right, so you can recognize their names. One's called glycine. Glycine's R group, seriously, its R group is nothing but a hydrogen? You couldn't do better than that? I might even ask you to memorize that one, right? That's a pretty easy one to put your head around, and it's just got an H dangling off of it. You cannot have a simpler R group than just putting a hydrogen there. It's like they're not even trying, right? That's just putting something there to fill out the valence for the carbon. There's not much else going on there. That is by far the simplest of all of the R groups. But because it's a nonpolar covalent bond, we put it into this bin with the other hydrophobic R groups. That's not too much more imaginative. A methyl group dangling off, it's a little bit better. You tried a little bit more than what happened over here, but nothing but nonpolar covalent bonds. Here, now we're getting a little bit more interesting. And you work your way down the line here, and notice how you have some that are really small, and some where the R groups, that's a pretty big boy right there. That's going to take up some space at a molecular level. It's going to cause other things to have to get out of its way. So we've got a bunch of different hydrophobic amino acids. I'll do a quick count. I'm thinking I see 11 hydrophobic amino acids up there on the screen right now, right? And they have a variety of different structures for their R groups. They all have nonpolar covalent bonds. Some are really teeny. Some are a little bit bigger. Some of them are kind of huge in terms of the side group that's dangling off of that amino acid. There you go. Those are the hydrophobic ones. If I showed you on a test an R group, and I said the R group was this methyl group here, and I asked you what kind of amino acid is it? Is it a hydrophobic, a hydrophilic, a positively charged, a negatively charged, or, I don't know, I'll make something up, anti-disestablishmentarianist R group, right? Yeah, I don't know, I gotta come up with something for a fifth choice. That methyl group, that means it's part of a hydrophobic amino acid, right? That's all you really need to get in your head to do well in this class for the rest of this class, including working on the first test. If you also happened to know that its name was alanine, I'd say good for you, but I don't think I'd have any way to pick that off of a multiple choice test. Right? I'm not going to be asking you which of these 5 structures corresponds to the R group for alanine. I'm not going to do that. All right? So that's what you have to look forward to here. If you can be comfortable with that, you're in good shape dealing with the rest of what I'll say about proteins and how they work. So those are the hydrophobic guys. A little bit more interesting. There are 6 that fall into this hydrophilic category. The big difference between the hydrophilic R groups and the amino acids that are associated with relative to the hydrophobic amino acids and the R groups that they have, the big difference here is that now, for the first time in this discussion, we're seeing some things that are other than carbon to carbon and carbon to hydrogen bonds. Okay? Specifically, we're seeing some hydroxyl groups and a couple of amine groups and aldehyde kind of things as well. All right? But check it out. Serine, about as simple as you can get for something that's hydrophilic. Check it out. You got a methyl group effectively over here, and then attached to it in place of one of the hydrogens is a hydroxyl group. That hydroxyl group is going to have 1 and then 2 polar covalent bonds. Water molecules are going to like to interact with those polar covalent bonds because of the separation of charge that they'll have associated with them. Hydrophilic, water-soluble, likes to interact with water. How many other ways do I need to say it? That's what's going on with these guys. So, serine, because of that hydroxyl group, is pretty recognizable as hydrophilic. Threonine, because of its hydroxyl group, pretty recognizable as hydrophilic. Cysteine, because of the sulfhydryl group, which is chemically pretty similar in its behavior to a hydroxyl group. Cysteine, because of that sulfhydryl group, pretty hydrophilic. Just as an aside, remember I talked with you about these functional groups, what was it, a week ago, I think, for the first time, and I told you sulfhydryl groups play an important role in cross bridges, disulfide bonds that link one part of a molecule to another part of a molecule or to a different molecule. When that stuff is happening in proteins, it's happening because of cysteines. One cysteine on an amino acid can make a covalent bond with another cysteine through those sulfhydryl groups. All right? Tyrosine, another amino acid, hydrophilic because of that hydroxyl group. The rest of this stuff is super hydrophobic, but you put that there and it makes it water soluble. Asparagine, this is an interesting case, right? There's no hydroxyl group here or here for either asparagine or glutamine, but check out that amino group, check out the carbonyl group there. There's separation of charges going on, maybe not full charges at neutral pH, given that structure, but there's still a separation of charge, and that's going to make it water soluble, hydrophilic, moving in the direction of not just being hydrophilic, but charged and seriously hydrophilic. Again, what do we see beyond that? Some of these guys are relatively small R groups. Some of them, tyrosine, that's pretty big. It's pretty bulky. Other stuff's going to have to get out of its way to make room for it when a protein gets put together with a tyrosine in it. And we're in the home stretch. There are 2 amino acids that are negatively charged at neutral pH. There are 3 amino acids that are positively charged in the normal environment for most of our cells. It shouldn't be a surprise to you to hear that for the negatively charged ones, they're negatively charged because part of the R group is a carboxyl group. So you can see aspartic acid and glutamic acid both have carboxyl groups associated with them. It shouldn't surprise you at this point to hear that for the positively charged ones, they're positively charged because they have at least 1 and sometimes 2 amino groups associated with them, and that brings with them a full positive charge. So, a fair bit of detail has just been thrown at you. At the end of the day, what's going to be really important for you to be comfortable with is that, again, we've got a spectrum of color, in some sense, a palette that we can draw from to make proteins that have different behaviors depending upon which of these bins we go into. Do we use glycine? Do we use tyrosine? Do we use aspartic acid? Right? Each of those, whichever of those amino acids we dial up to link together to the next spot in a growing biopolymer, a protein, whatever we dial up is going to have something to say about how that protein works, how it behaves, what it likes to hang out with, all of that sort of thing. And we've got 20 shades of gray to work with here. The most important axis to keep track of when you're talking about these things is hydrophobicity versus hydrophilicity, how water-soluble the R group is versus not water-soluble, until you get way to the end of the spectrum, and we're talking not just about water-soluble, but seriously chemically reactive because it's got a full-blown negative charge or a full-blown positive charge, right? So that's the most important axis, the most important continuum to worry about. But also on that is this other dimension of how bulky it is. You've got small amino acid R groups, you've got big amino acid R groups. That's also something else to keep in mind. But let me tell you something that kind of blew me away. Might not mean that much to you right now. But if you reached into your proteins, the proteins that are doing what you need to have done within your body, and you replaced a relatively small hydrophobic amino acid, go back and call one out by name, you replaced an alanine with a glycine, right? If we could reach in, if God could reach in and hold his hand over you and say, all of your alanines, poof, are glycines right now in all of your proteins throughout your whole body, you know what would happen to you? Probably not that much, right? Pretty much every protein, wherever you have a glycine, if you substituted it with an alanine, there are going to be a handful of enzymes where you really needed to have an alanine there. That was the key amino acid that made that protein work, a couple. But for tens of thousands, maybe hundreds of thousands of different proteins, you would not be able to determine that substitution had been made. The enzyme, from all of the tests we could perform on it, it would have the same shape, it would have the same chemical reactivity, nothing would be different. Largely interchangeable when you're talking about amino acids that have similar basic characteristic in terms of hydrophobic versus hydrophilic. Now, if you substituted all of the alanines with one of the hydrophilic amino acids, threonine, better yet, serine, not that different in size, right? The size is about the same, but I put a serine there instead of an alanine, that might make you a little bit sick, right? That might cause you some problems. But if you do substitutions within a group, and especially if they're similar in size, probably doesn't have much of an effect. They're largely interchangeable. But again, we got a continuum. We got this whole spectrum to work with here. And if you can appreciate that those amino, that's the key. Those are the features of the R groups that are functionally important. I think you can kind of then piece together all the rest of what makes proteins tick just by keeping your eye on that particular parameter. So hydrophobic, hydrophilic, negatively charged, and positively charged amino acids. And I've been alluding to it all along, but you link these amino acids together one after another in a polymer, and those polymers are called proteins. The monomers that get strung together are the amino acids. The bond that gets made between one amino acid and the next amino acid is called a peptide bond. Those bonds are made through dehydration reactions, and they're broken to get back to the individual monomers through a hydrolysis reaction, just like we were talking about with the carbohydrates and to some extent with the lipids. Same old story. So the bond between the carboxylic acid of one amino acid and the amino group of the next amino acid in the chain is a peptide bond. And here's a bit of trivia for you. When you're making a protein, if you look at the structure, the chemical structure that's going on, notice that one of the amino acids at the amino acid at one end of the molecule is still going to have this unengaged, sort of naked amino group. All right? Guess what we call that end of the protein? That's the amino end of the polypeptide. It's the amino end of the protein, and it gets its name, and it's sometimes referred to as the N-terminus, just drawing attention to the nitrogen in the amino end. It gets its name because that's still dangling open, free at one end of the protein. At the other end of the protein, no matter how long it is, if it's 500 amino acids long, at one end you'll have an amino group. At the other end you'll have this carboxyl group, the carboxylic acid, sometimes referred to as the C-terminus, the carbon end. All right? So that becomes important because when cells string together amino acids, they use ribosomes to catalyze that reaction. When cells string together amino acids, they always start at the amino terminus, and they always end at the carboxy terminus. New amino acids always get added on to the carboxyl end of a polypeptide. All right? And so to a molecular biologist like me, I'm thinking, what's at the beginning of the protein? And when I'm thinking what's at the beginning, I'm thinking in fancier terms perhaps, what's at the amino terminus? What was the first amino acid that started the chain that got polymerized out to make us this big old protein down the line? So one amino acid gets hooked up to another amino acid by a peptide bond. That made us a dipeptide. If you bring in another amino acid to make this dipeptide into a tripeptide, you do it with a dehydration reaction. The water gets kicked out. And whereas here we have a dipeptide, here this is a tripeptide. After 4 or 5, nobody counts the peptides anymore. They just say it's a 500 amino acid long protein or a 322 amino acid long protein. Okay? So there you go. That's the polymer part of proteins. I can't wait to tell you now a little bit about what we do with these proteins. These are best guesses, maybe a little more than a guess, but this is the way that proteins fold up on themselves and have been, the shapes of these proteins have been inferred through a number of different means. We have a pretty good grip on how it is that proteins fold up. The thing is this, I mentioned it on Tuesday. I'm going to mention it more forcefully today. It's going to come up time and again throughout the rest of this class. In biology, at all levels, not just at the molecular level, not just at the cellular level, not just at the organ level or the organism level or even the ecosystem level. In biology, time and again, over and over, you're going to hear people telling you that structure equals function. There's an equating between the shape of something and the way the thing works. If you're trying to figure out what something does in a biological setting, figuring out what the function is, your best clues almost always come from figuring out, well, how's it put together? What's its shape? That is so unequivocally true for proteins. The structure of a protein, much more than anything else, dictates, governs how it is and what it is that protein does. And I'm going to come back and I'm going to give you examples of that over and over again many times during the course of this class. And I'm going to encourage you often to think, what if you did this to change the shape of this protein? How do you suppose its function might change? So when I tell you we string together these amino acids to make proteins that are hundreds of amino acids long, that can't be, and it is not, the end of the story. How it is those strings of amino acids fold up on themselves is ultimately what's going to determine how it is that protein works. All right? So being able to point to a structure of a protein is an important aspect of molecular biology, and often, very often, almost surprisingly often, I get surprised at how often it's the case when somebody tells you the kind of life I lead. People show me the structures of molecules, of protein. Hey, check this one out, right? You know, people show me the structure. I'm surprised at how often I don't need anybody to tell me, oh, you know what? The action is going to be right here, right? I mean, the active site of enzymes, where the catalysis takes place, where the enzyme is catalyzing something, usually just hits you right between the eyes. I mean, it jumps out at you. You don't have to be particularly well trained to say, there's something happening around over here. And look at that. It's like the whole rest of the molecule is kind of there just to set up that little groove, that little pocket that something else can fit into and get chomped on or tweaked in some way by whatever it is that this enzyme happens to be. All right? So structure and function, hugely important for proteins, important for pretty much all of biology. So how do we get from a string of amino acids to things that fold up to give us these structures so regularly, so reproducibly? Well, let's take it step by step. There are 4 levels at which we'll talk about proteins having structure, right? And I'll just rattle them off for you here. The names aren't particularly imaginative. There's the primary structure, the secondary structure, the tertiary structure, and then the quaternary structure. 1, 2, 3, 4. 4 different levels of structure. All right? The primary structure is pretty obvious. It is the linear sequence with which amino acids got strung together for that particular protein. That's the primary structure of a protein. It is simply the order in which the amino acids got dialed up and added to the growing polypeptide. What protein is that? Darn, they don't. Oh, yeah, it's transthyretin. All right? So that's the protein that you see up there. This transthyretin apparently is 127 amino acids long. It has a beginning, it has an end, and it's got 127 different amino acids all linked together. I presume this is human transthyretin. Not particularly important for what we're talking about here today. Could be rabbit transthyretin. Doesn't really matter. But here's something that might matter to you. It's been a point of confusion with some students in the past. Every one of your human transthyretins will have exactly the same primary sequence, exactly the same primary structure, exactly the same order in which amino acids got strung together. All right? You also make other proteins aside from transthyretin. Oh, I don't know. Insulin is one people talk about a lot and know about. Insulin is a different protein, and guess what? It has a different number of amino acids. My recollection is it's 50-something. Not important. And it's got a different sequence of amino acids. All right, so every one of our different proteins has a different primary structure, and that's because they're different proteins, and that makes them ultimately fold up and do different things. So if you need a protein to do what transthyretin does, I happen to know that the best way to fold it up to get to do that thing is to have that particular sequence of amino acids. Anything else will probably be just a little bit less good at doing what it is we need transthyretin to do. Right? It's a real credit to the power of selection to keep us all in line and make sure all of our transthyretins are up to snuff. We probably all, if it's an important protein doing an important job, we probably all have the same amino acid sequence. But I can guarantee you this, the amino acid sequence, the primary structure of transthyretin is certainly different than the primary sequence of another protein like insulin. Let me commiserate with you here. I'm looking for some sympathy here. I have a very vivid recollection. The reason that insulin popped into my head just now, I have a vivid recollection of my time as a graduate student. I was a biochemistry major in a biochemistry PhD program. In the first semester of graduate school, some jerk, and I can picture him, I won't mention his name, I know his name still, just because he did this to me, he made me memorize the amino acid sequence for the insulin protein, for the human insulin protein. It's like, seriously? The internet was around back then, right? I could have looked it up whenever I needed it, but no, you can't have access to the internet during my exams. You've got to memorize the amino acid sequence of insulin. To this day, I can't figure out what purpose that served, aside from giving me a story. I think it was sort of like a hazing sort of thing. You want to be a PhD in biochemistry? Well, we have to have shared adversity. I had to memorize the structure of insulin. You have to memorize the amino acid sequence of insulin, right? I'm not going to make you guys do that. It stops now, right? No more of memorizing the structure of insulin, the amino acid sequence. But sadly, there are people who do that sort of thing. So that's the primary structure. It's simply the sequence in which amino acids get linked together. And what does that do? Here's what that does. It gives rise then to a secondary structure. Certain strings of amino acids have a natural tendency, because of the size of their R groups, because of the hydrophobicity of their R groups, they have a certain tendency to just fold up on each other in particular ways. That's secondary structure. Secondary structure, in turn, inspires tertiary structure, and tertiary structures give us quaternary structures. Here's some blown-up examples of secondary structures. There are two big ones that account for 70%, maybe 80% of a typical protein's shape. All right? There's the alpha helices and the beta sheets. Alpha helices, shown up at the top there. You can see how we've got amino acids, one after another, strung together, with their R groups dangling off of them. They fold up into these coils. And I don't know that there's much purpose anymore to it, but a telephone cord is like the world's best example ever of an alpha helix. Some of you have probably seen telephone cords before. That's an alpha helix. If you need to hold in your hand something that's an alpha helix, go find an old style phone with a cord. That's an alpha helix. The amino acids, because of their natural inclination, their size, their charge, or their lack of charge, they just fold up and they make these coils. We call those coils alpha helices. All right? And then there are other strings of amino acids that are most comfortable when they fold up into these pleated things called beta sheets. Telephone cord, best mental image I can give to you for an alpha helix. My daughters would cringe, but I'm going to give you a mental image for a beta sheet. An accordion pleat, right? You know, one of my daughters is playing clarinet in school, another's playing bassoon in school. You know what I played in school? I grew up in that part of the world where it was popular for kids to play accordions. So, you know, maybe someday instead of playing a YouTube video, I'll just bring in my button box accordion. We'll have a little dance down here, do some polkas, some waltzes. It'll be great. But those beta sheets at the protein, the secondary structure of proteins, beta sheets are for all the world, look like and act like. They expand and contract just like accordion pleats do. Okay? So those are secondary structures. Again, they fold up on themselves. You don't need to have somebody reaching in to do this. You don't have to have some sort of funnel they pass through. It's just because it's intrinsic to the amino acids themselves. In certain sequences, they like to adopt these two different shapes. Secondary structures. And then, beta sheets are often shown as these flat ribbons when you're looking at these structures. Alpha helices are shown as these coils. Then, when you put together, you get your amino acid sequence, the primary structure. It inspires a certain set of alpha helices and beta sheets. Those tend to fold back on themselves to then give you this three-dimensional structure that's called the tertiary structure. Okay, so folds up like this. Once again, you're getting a good look here at transthyretin. And when one protein interacts with another protein, we start talking about quaternary structures. Transthyretin, it turns out, works as a dimer, or here, better, as a tetramer. Apparently, you need to have 4 of those individual transthyretin proteins come together to interact with each other in a certain way to give you the functionality that we associate with transthyretin, whatever it is that transthyretin does. So, primary structure is the amino acid sequence. Secondary structure is the way that primary structure inspires alpha helices and beta sheets. Tertiary structure is that overall three-dimensional configuration that you get after that. And if the protein interacts with other proteins, including other versions of itself, but it could be other proteins entirely, those interactions are called quaternary, and they give rise to quaternary structures. All right. And here's another protein that you've probably heard about, hemoglobin. Hemoglobin is another tetramer, so you've got this bluish. Somebody help me out. What kind of color would you say that is? Aqua? Cyan? Blue. Okay, so you got you got this bluish alpha helix over here. Oh, I'm sorry, that's the bluish beta subunit. You got one beta subunit, a second beta subunit, and then 2 purple alpha subunits. So hemoglobin that carries oxygen in your blood is actually a tetramer. You've got 2 copies of alpha globin and 2 copies of beta globin that come together to give you the quaternary structure that gives you the functionality of hemoglobin. Each of those 4 subunits carries with it 1 heme molecule. Those are the things that are actually physically interacting with the oxygen in your blood. But you can see at a glance the structure, the physical shape of hemoglobin is different than the physical structure of transthyretin. They're doing different things. Structure, function. One and the same in a biological context. There's one more thing. From what I've just been telling you, you might think that if you give me the primary structure of a protein, that I can do some magic, probably using a computer, to tell you, here is the tertiary structure, here is the quaternary structure, this is what the protein does. All I can say is, I wish that was so. Turns out, one of the grand challenges, and it's referred to as a grand challenge for molecular biology, is being able to go conceptually from that primary sequence to be able to predict what the quaternary structures will be. If we could do that, we could solve so many disease problems. We could create proteins from scratch that did things that we really wanted to have done, like binding atomic gold from seawater and putting it into nice, neat little bars that make us each individually rich, right? I mean, there's so many things that you can do. We know proteins could do those things. We can't quite figure out how to put it all together yet. Hemoglobin always folds up just the right way inside of all of our cells. We can't quite figure out how you get from the amino acid sequence to that final structure yet. It's a grand challenge. We're pretty good these days at going from primary to secondary structure. And we're not as good, but pretty good at going from secondary to tertiary. And we're good, but not very good at going from tertiary to quaternary structures. So there's a lot of work that still needs to be done. We know it happens. It happens billions of times a second in our bodies. Proteins fold up in just the right way. It works at an atomic, at a molecular level. We just don't seem to know what all the rules are just yet. And so people are very actively working on solving that kind of problem. What I've just described to you in a nutshell there is one of the biggest problems facing a discipline called bioinformatics. Wright State has a couple of really neat courses on bioinformatics. I'm the lead author on a textbook that we use in a couple of those classes. Those are the sorts of problems that bioinformaticians often wrestle with. So we know the theory, we just can't quite work out the practice of it yet. And we know the importance of the problem. It's just, again, we can't quite get from what's stored at the level of DNA to what's actually getting stuff done inside of all of our cells. So I just said it, you know. That's a nice sort of segue. What's stored at the level of DNA? Let's talk about the fourth of these four biopolymers, nucleic acids, DNA/RNA. Carbohydrates and lipids are where we store energy. Proteins are what we use to physically get stuff done. They catalyze reactions. They give us structures. They move stuff around. They break things down. Nucleic acids are what we look to as living things to store all of our heritable information. And so that's the answer to question number 5 in the pre-quiz that I gave all of you. Let's see how you guys did. A little more than half recognized right out of the gate that nucleic acids, and I think what tripped you up, if I had said DNA, I bet you most people would have agreed that it's DNA that stores information. As a biology professor, I have to be inclusive, right? So it's not just DNA, it's also RNA, but for all intents and purposes, it's DNA, right? So that's why I said nucleic acids instead of DNA. But in terms of the basic functions of these biopolymers, most of you came into this class already knowing nucleic acids are the thing where information is being stored at. And this is a picture I'm going to show you later in the course. It's essentially just a convenient way for me to talk about the central dogma of molecular biology. Again, that's going to be a topic in about 2 weeks that I'll talk with you about. The long and the short of it is that within eukaryotic organisms like you and me, we've got a nucleus in all of our cells, okay, most of our cells. We have a nucleus in most of our cells. That's the problem with it. There's so many of you. I know there are people out there thinking, ooh, what about red blood cells? They're anucleated, right? But anyway, so there are some exceptions, but most of our cells have a nucleus, and it's within that nucleus that the genetic instructions are stored, and they are stored in the specific linear sequence with which 4 different nucleic acids, 4 different nitrogenous bases get strung together. So all of this is pretty localized to what's going on here in the nucleus. The information comes out of the nucleus in the other kind of nucleic acid, the RNA molecules, and that gets used by ribosomes to string together the amino acids that we need to make our proteins. So that's the central dogma. We'll talk more about that later. Let's talk today, in the last few minutes that I've got, about nucleic acids and nitrogenous bases. Okay? Nucleic acids store information. That's their purpose, that's their function. And they store information in these biopolymers very simply by the linear sequence with which nucleotides get strung together. So this is a biopolymer. The unit, the monomer that gets linked together to make a long chain is called a nucleotide. There are only 4 different kinds of nucleotides that are used to make DNA molecules. There are 4 that get used to make RNA molecules. There's overlap between the two. Let's not worry too much about those details right now. But we're storing information, and the information gets stored by the linear sequence with which nucleotides get strung together. There are only 4 nucleotides that we use to make our DNA molecules. They are between friends known as C, T, A, and G. You are only trying to impress people, and it's obvious that you're just doing it to impress when you talk to some other molecular biologist and say, oh, gee, I got a lot of guanines going on here today. They're interacting with a lot of my cytosines, and I don't see a lot of action happening with my adenines or my threonines, but my guanines, they're doing great. Nobody talks like that. All right? We all just say to each other, hey, that G wants to base pair with that C, doesn't it? Right? It's always G and C. It's always A and T. It's nice that you happen to know that the G is shorthand for something else, guanine, and that the A is shorthand for something else, adenine, and the T is shorthand for something else, thymine, and the C is shorthand for cytosine. And when we're talking about RNA, instead of the T, the U is short for uracil. Right? So those 1-letter abbreviations are that. They are abbreviations. But typically, you'll hear people talking just about G's, A's, T's, C's, and U's. And when you look at a nucleotide, you'll see 2 features are always the same, and 1 can be different. The features that are always the same, let's start with the sugar. It's a 5-carbon sugar, a pentose sugar. You've seen these structures a couple times now in this class. That's one of the features that's always there. Another feature that's always there is there's a phosphate group. And then the 3rd feature of a nucleotide is this thing that differs from one nucleotide to another. In amino acid terms, we're talking now about the R group. You don't talk about it in quite that same way for nucleotides. But there are 4 different nitrogenous bases that you can use for DNA and 4 that you can use for RNA. Those nitrogenous bases, this variable part of the monomer, are C, T, U, A, and G. Okay? So I looked it up. We've got 26 letters in the English alphabet. Right? I'm feeling good about that today. There have been times I've thought it was 24. Today I know it's 26. All right? There are 4 letters in our DNA alphabet. We can string together letters to spell things out that have specific meaning. There's information associated with the way we string letters together. It's the exact same principle for stringing together the 4 letters that we use for DNA. Because there are only 4 letters, DNA words, things with useful chunks of information, need to be longer. Pretty much what it comes down to. And we string together hundreds, thousands of nucleotides to spell out the instructions that you need to make every protein that we use. That's where the information is being stored. It's being stored on that kind of scale. Hundreds, thousands of nucleotides linked together spell out what a ribosome needs to know to be able to make a particular kind of protein with a particular sequence of amino acids. Your genome is a prodigious genome. It's a big one. You should be proud, right? Your genetic information from beginning to end, 3.2 billion nucleotides strung together in a particular way to make you most of what you are. 3.2 billion. If you wrap your head around how much information that is, that's the equivalent to about 32 complete sets of encyclopedias, right? The same equivalent amount of information. You need about 40 compact disks to store the information that corresponds to the 3.2 billion nucleotides that is the human genome. And again, ultimately, all of those nucleotides have a phosphate group and a sugar and a nitrogenous base. It's which nitrogenous base, the sequence, that determines that information. I'd like to leave you with, I think, one last thing, right? I didn't make you memorize the structures of the amino acids, right? I'm a pretty cool guy that way, right? Maybe I should balance things out and say, but by golly, memorize the structure of the nitrogenous bases. I'm not going to do that, right? But here's something I'd like for you to be able to do. I'd like for you to be able to distinguish between the purines and the pyrimidines. There are two different basic kinds of nitrogenous bases. There are the ones that have one ring. They are called the pyrimidines. And then there are the ones that have two rings. Those are called the purines. And I'll tell you very frankly how it is I remember which is which, right? Purines with their two rings should have the bigger name. They don't. All right? There you go. That's my secret for figuring out the difference between a purine and a pyrimidine. We string them together by making polymers. We link them one monomer to another with a phosphodiester bond, and you end up then with structures like this double helix that contain all of that information. And then I've got a couple last slides that are part of the PowerPoint presentation for the class. I've just slipped them in. I don't want to talk about them, but I've slipped them in at the end of the PowerPoint because it seems to me that they very succinctly tell you some of the more important features of each of these different kinds of biopolymers. If you want to compare what you've written down to what I've been saying, these might be a good thing for you to look at just to make sure you've got all these structures down and you know what's going on. Have a great weekend. See you guys on Tuesday.